Okay, so let's talk about photosynthesis. Where does, number one, what kind of organisms does photosynthesis occur in? Plants and some plankton, okay? But where, what is the organelle? Chloroplast, okay? So, this happens in chloroplast. Okay, now let's talk about what a chloroplast looks like. Alright? There are 3 parts to a chloroplast. Alright, so you're going to have the chloroplast membrane. Okay, so the chloroplast membrane basically just allows nutrients in and out of chloroplast. And what's the word I want to use? Closes everything in. Okay? So that's the chloroplast membrane. And as we're going, okay, so chloroplast membrane. Okay? Now, the next thing that we're going to talk about is inside of the chloroplast, there are these little discs. Okay? So there are discs called thylakoids. Okay? So thylakoids, small discs inside chloroplast. Okay? Now thylakoids are the thing that break apart water. Okay? And so I draw a stack of them. Okay? It's a stack of thylakoids. Not yet. Alright, so a stack of thylakoids come in stacks. And these stacks are called grana. Grana. Okay? So we've got thylakoids and we've got grana. So, and then the third thing we want to talk about is this stuff called stroma. Okay? So, like cytoplasm is the gel that kind of surrounds everything inside your cell, stroma is essentially the gel that surrounds everything in here. So this is stroma. Okay? This is the gel surrounding thylakoids. Okay? And this is where glucose is produced. Okay? Are there any questions about that? Just really fast. Is this guy chlorophyll? I know we kind of mentioned it yesterday, but what does chlorophyll do? Not yesterday, Monday, but takes in Take a guess. There's like 3 things that go into
Oxygen.
not oxygen. 2 other things.
Water.
Not water.
Sugar.
We're making
Light.
it's sunlight. Okay, so this is a pigment that is used to capture sunlight. Okay? And we use that light energy to break apart the water in our light-dependent reactions in your thylakoids. Okay? So chlorophyll captures sunlight. What else does it do to a plant? What's that?
Makes it green.
Why? How does it make it green? So you guys know what that stands for?
Colors of the rainbow.
Colors of the rainbow. It's also the colors of the visible light spectrum. Red, orange, yellow, green, blue, indigo, violet. Okay? What do you think, which one of those do you think chlorophyll captures and which one of those do you think chlorophyll doesn't capture?
Green.
Green.
It captures.
So we think it captures green?
It doesn't capture.
No,
So chlorophyll collects all colors except green. Okay? It absorbs them. I guess I should say absorbs, not collects. Absorb is a much better word. So what does it do to green if it's not absorbing it? It's reflecting it. Alright? It reflects green, which is why we see the green color. It reflects the green waves. Okay? Are there any questions about chlorophyll? This equation. Photosynthesis has an equation that looks like this. So what 3 things do we need to do photosynthesis? You're going to need some sunlight, okay? Plus what? We need water, okay? And what else goes into it? Not oxygen. Plants don't need oxygen. CO2. Okay? Now, using those 3 things, what are we going to make? We're going to make 2 things. Energy in the form of glucose. Okay? And glucose, C6H12O6. Okay? Yeah, you say you're going to write glucose, but you're not going to write glucose because that's like one of the few things that you're going to need to know.
Oh, really?
Yeah.
Oh, okay.
You can write glucose above it, but you need to know that glucose is C6H12O6. It's the most important sugar in the world. Pretty much everything runs off of glucose. Well, every organic thing runs off of glucose. Okay? And what else do plants make? Oxygen. Okay? Now, is that balanced?
No.
No.
No, right? I only have 2 H's over here and I have 12 over here. What should I do to make them balanced? Slap a 6 in front of that. Right? So now I've got 6 times 2, 12. How many carbons do I have on the glucose? 6. How many do I have on CO2? What do I need to add in front of it? 6. Right? So now my carbons and my hydrogens are all equal. But I have 6 plus 12. How many do I have over here? 18. And right here I have 6 plus 2 is 8. So removing 6 slap a 6 in front of this. So now we have 6 plus 12 is 18, okay? So in order to make 1 glucose, you need sunlight as energy, you need 6 water molecules and 6 carbon dioxides. And in the process, you'll make 6 oxygen molecules, okay? This is the photosynthesis equation. And you don't need to write this in your notebooks yet, but when we start talking about cellular respiration next week, which is what we do and plants do to actually use the glucose to make energy, you want to see what that looks like? You're going to take C6H12O6 plus, what do humans need to breathe? Oxygen. So you're going to take glucose and oxygen. And guess what humans make when you breathe out? What are you breathing out? You breathe out carbon dioxide. Whoop. I would know this equation. I'd know both of those equations eventually, but know that one. Right? If I ask you on a quiz, how many carbon dioxide molecules are needed to make one molecule of glucose? The answer would be 6. Now, if I asked you how many molecules of oxygen are made when you use 12 water molecules, the answer would be 12, because it's 6, 6. So those are always the same. Okay? All right. Let's talk about the more confusing part. So we've gone through the basics. Now we need to go through the specifics, okay? The actual details of how these things work. And the first thing we're going to go through is the light-dependent reactions. Okay, so with the light-dependent reaction, this is going to be where we break apart water using sunlight, and we're going to get the things that we need in order to produce glucose in the second one. All right? So, this happens in thylakoid membrane. Okay, so it happens in the thylakoid, but it mainly happens in the membrane of the thylakoid. So I'm going to kind of draw a picture and draw steps that go along with it. All right? And so this is going to be the start of our membrane. All right? This is the outside of the thylakoid, and this will be the inside. Okay, so outside, inside, membrane. All right, so what we're going to do first is we are going to run into this molecule. All right, the first thing is we use this molecule called Photosystem 2. Okay? Photosystem 2. And what this is going to do is it's going to take in light, energy. Alright, so we've got our light energy. And the next thing that it's going to do is break apart water. So you'll have H2O. It's going to go into here, and it's going to become H plus, H plus, and then O minus. Okay, it's going to break it all the way apart. Everybody good there? So we've got a bunch of hydrogen ions and some oxygen ions. Now the other thing that this does is it is going to remove an electron. Okay? So we're going to have an electron, as I'll just put as E minus, okay, that will enter the membrane. And so throughout the membrane, the electron is going to travel, all right? And so here, let me put this as like numerical steps. So step 1, photosystem 2, uses light energy. Go ahead and check your things. This. Okay, so photosystem 2 uses light energy to break apart water, H2O, okay? Electron is released into membrane. Okay, now we're going to go from photosystem 2, all right? What do you think we're going to go to? You'd think that it would make so much more sense if it did that, but we're actually going to go to photosystem 1. Why it's named this way, I don't know, but that's the way that it's named. What comes after 1?
3. Don't say 3.
Ferredoxin. Okay, so, and again, why it does this, I don't know. It's a weird thing. But so step 2, as electron travels from photosystem 2 to photosystem 1, the charge allows hydrogen ions to move into the thylakoid. Okay, so hydrogen ions come from the outside in. Okay, as this thing moves from there to here. All right? So, once we hit photosystem 2, this is where we make something pretty important for the next step. Okay? Photosystem 2, I'm going to draw this. I'm going to draw that. But at photosystem 2, the electron. What? Photosystem 1. Ugh. 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 Yes. Okay, so the electron will eventually leave the membrane. Okay? But, so step 3, at photosystem 1, electron energizes molecule and combines Na plus and hydrogen to form NADPH, which will be useful in the Calvin cycle. Okay, we'll see why that gets used later. But anyway, so we'll put NADP plus hydrogen. That goes in there, and then out you get NADPH. That's what's known as an electron carrier molecule, and we will discuss why that is important in a little bit. And then really there's a third step, or fourth step, which we will do as soon as you guys go check your things. So, the final step, we use this enzyme right here. This is called ATP synthesase. Okay, so ATP synthesase. Now,
Why did it go to photosystem 2 to photosystem 1 to ATP?
well, we're not going to ATP synthesase.
Oh.
Right? That's just something that, it went from photosystem 2 to photosystem 1. And that's what the electron was doing. This does something completely different. But it's utilizing things from that. Okay? So right here, when we broke apart water, we got 1 oxygen and 2 hydrogens, right? So we've got those. And then as it was going across, we've also brought in more hydrogen, right? So what do we have a lot of inside of the cell, or inside of the thylakoid? Hydrogen, okay? So we have a huge hydrogen, H+, gradient inside thylakoid. Okay, and that's important. There's a name for there being a huge gradient, and it doesn't like this, all right? The name for that is called chemosis. Chemosis, okay? So chemosis is when you have a really big gradient of hydrogen. We want to get that hydrogen out, right? When you have a big gradient inside, you usually want to push it to where there isn't a gradient. And that's what we use this guy for. Okay? So you guys know how like a hydroelectric dam works? When water comes through, it turns a turbine and it makes electricity, right? This kind of works the same way. We've got all of this hydrogen and it pushes through here, okay? And so we're putting hydrogen from the inside out. But as it goes through there, it essentially is turning it, creating energy. And that energy that gets created is going to make what? ATP, okay? So we will use this, and I'm just going to draw ADP coming out as ATP. Okay, and so step 4, which I'll put right here.
Running out of room.
I know. This is the last step, so. Alright, so step 4, hydrogen, H+, moves through ATP synthesase and creates ATP. That's the simplest way I can put it. So basically, these steps go: light breaks apart water, okay? Removes electron. Electron moves from photosystem 2 to photosystem 1, pulls hydrogen across. Step 3, electron energizes photosystem 1, creates NADPH. Step 4, because of the large gradient of hydrogen, or chemosis, it moves through ATP synthesase. As it moves through, ATP is created. Okay? That's kind of the short and sweet version of it. Still, this is one of the more, I'll say, confusing parts. Alright, the last part, second part, called the Calvin cycle. Okay? So during the Calvin cycle, we are going to take 6 CO2. Okay? So our 6 CO2 are going to come in, and they are going to mix with it's just a bunch of weird carbon molecules, okay? And we'll tell you where those molecules come from here in a little bit. But so they're going to mix, and they're going to break apart, and you're going to make these things called, you're going to make 12 of them, and they're called, God, is it G3P? No, it's 3PGA. Ugh. 12, 3PGA. Alright? And what 12, 3PGA means is that you get 12 of these 3 phosphoglycerate molecules. Okay? But the thing to know about these is that they have 3 carbons. Okay? So they each have 1, 2, 3 carbons. Alright? Now we're going to use, remember when we made that NADPH and the ATP in the light-dependent reactions? We're going to use some of that. Bless you. And we're going to make, instead of the 3PGA, now we're going to make these things called G3P. Okay? So we'll make 12 G3P. Which stands for glyceraldehyde 3-phosphates. Okay? Okay, this is also a 3-carbon molecule. Okay, it's just a different one. It's got a different structure, and we need that different structure. So we'll have 3 carbons. Okay, however, we're going to use 12 NADPHs. Okay, and those will become 12 NADP plus. Okay, we're going to use that hydrogen. We're also going to use the energy from 12 ATP, and you'll be left with 12 ADP. Okay, breaking off that last phosphate. All you're going to have there is 12 ADP and 12 NADP plus. Okay, so it takes a lot of energy to actually end up making those G3P. And those G3P are important because those are what we actually use to make glucose. Okay? So, we're going to go through this process. I'm going to draw this. I'm also going to draw this. Okay? So right here, 2 G3P leave. Okay? So they're going to leave the cycle. Now, how many carbons are in G3P? 3 of them, right? How many carbons are in glucose?
Well, 10 now.
No, in glucose, C6, right? C6H12O6. We're going to take these G3P. We also use some hydrogen from here, right? To make these. So we have these 2 G3P leave and become glucose. Okay, there's another thing that kind of happens to them outside of this, but all you guys really need to know is that right here, these 2 are going to leave and they're going to become glucose. Okay, which means we're left with how many G3P? 10, right? Because there was 12 originally. So 10 G3P. We're going to take those and we're going to make this thing called, I always forget, ribulose 5-phosphate. Okay, and this one you don't need to know as much. Ribulose 5-phosphate. Okay, and we're going to have 6 of those. Now, ribulose 5-phosphate is a 1, 2, 3, 4, 5 carbon molecule, correct? Yeah. Okay, so we've got 5 carbons. Now, we need to turn this from ribulose 5-phosphate into ribulose 1,5-phosphate, again, which is what we call RuBP, okay? RuBP. But in order to make RuBP out of your ribulose 5-phosphate, you're going to again need 6 ATP that get used up and become 6 ADP. All right? This right here is what combines with the CO2 to make this whole cycle start again. And in order to do that, we need this enzyme called Rubisco. Okay? And Rubisco takes RuBP and combines it with CO2 to make 3-PGA. Okay, so basically all this really says is that we're taking CO2 and through a long process, okay, we're going to use energy to produce glucose, and then some other carbons will be recycled so that the process continues to go. Okay? Let me write that in here. So, Calvin cycle, simple explanation. Okay? CO2 is used to make glucose. Okay? During process, ATP and NADPH get used. Okay? Several carbons are recycled to continue process. Okay, you don't necessarily need to remember all that stuff. If you remember that stuff in the middle, you'll probably do okay. Alright? You don't necessarily need to know 10G3P get used to make RuBP, okay? But do knows that we basically need to take carbon dioxide, 6 of them, using the NADPH and the ATP that we made in our light-dependent reactions. We're going to make 1 molecule of glucose. Okay? I give you more, but again, I'm going to simplify it into what I think, if you know, it should be good enough. All right? Are there any questions about this? Okay, I'll give you guys a little break.